Hi, my name is Taarun Ganesh, and I'm a junior at the Thomas Jefferson High School for Science and Technology in Alexandria, Virginia. I just want to start off by thanking IYRC for this amazing opportunity to present my project. So, my project revolves around an in silico design of a multi-epitope-based vaccine against the rotavirus. So, what is the rotavirus? The rotavirus is a mosquito-borne single-stranded flavivirus that initially appeared in Brazil in 1975, and it caused severe headache, encephalitis, vomiting, and blindness, among other symptoms. And in the 465 infections it caused, 20% of those patients were affected neurologically, and there's also a 13% case fatality rate. However, the rotavirus is still prevalent in today's society, as just more than a decade ago, in the 2011 dengue virus outbreak, the rotavirus reemerged, and many of the patients that tested positive for the dengue virus also tested positive for the rotavirus. However, there's still no licensed vaccine available for the rotavirus, even though serological studies in South America indicate that Brazilian horses are circulating that rotavirus, which means that there can easily be another variant of the rotavirus that emerges in today's world, similar to the COVID-19. So in my project, we took the rotavirus proteome that was available online in the National Center for Biotechnology Information bioinformatics server. So we took the 4 structural proteins and the 7 non-structural proteins, and we're able to use those structural proteins, which are so crucial for replication of the rotavirus within the host, and use it to create our computational vaccine candidate. So how do we create that candidate? We predicted epitopes. So an epitope is a part of an antigen where an antibody or a T cell receptor can bind to the antigen, triggering an immune response within the body. So in terms of predicting epitopes, we predicted 3 different types of epitopes. We predicted cytotoxic T cell, helper T cell, and B cell epitopes. So the reason we chose this multi-epitope based vaccine design as opposed to alternatives like the live attenuated vaccine is that the live attenuated vaccine poses the risk of reversion, where it recovers its virulence, whereas the multi-epitope based vaccine design is much safer and stronger. And since the epitopes aids in that stronger in that efficacy of the vaccine candidate. So in our project, we used 2 different types of vaccine delivery platforms that we'll get into later called the E. coli and the M13 bacteriophage system, which are 2 vaccine delivery systems that prove to be very cheap and affordable to use and develop further in the wet lab later. So going back into more of a methodology aspect, from the NCBI server, as I mentioned before, we retrieved that rotavirus polyprotein sequence. And using that polyprotein sequence, we screened each of those non-structural and structural proteins for antigenicity. And using that antigenicity prediction, we chose only the antigenic proteins to predict those B cell and T cell epitopes. And those B cell and T cell epitopes, they went through an extensive screening process consisting of antigenicity, its ability to elicit an immune response, allergenicity. Of course, we want it to be a non-allergenic, so it does not cause an allergenic response within the body, and non-toxic. So among many different types of biophysical properties tested, those were the 3 main categories we tested each different peptide sequence for. So this is where my project kind of splits into 2, with the M13 display and the E. coli system. So let's dive a little bit more into the M13 display aspect. So with the M13 phage display, we specifically use the P3 protein to fuse together our epitopes. So the P3 protein of the M13 bacteriophage is widely used as a fusion protein for all different kinds of peptides. And since it's able to display these large proteins and large epitopes that we selected this polyvalent approach, since able to produce and bind more proteins as compared to the monovalent display. So using each different class of B cell, T cell, B cell, cytotoxic T cell, and helper T cell epitopes, we're able to fuse those epitopes together with this P3 protein to create our M13 phage display. However, on the other side, with the E. coli system, as I mentioned before, we tested each of the structural and non-structural proteins for antigenicity, which elicited us to predict peptide sequences or these epitopes. So with each of the sequences that you see on the screen right now, we fuse them together with different types of linkers. So these linkers are specifically designed for these types of epitopes to fuse them together to create this final vaccine candidate. So our final vaccine candidate, using the Vacciden 2.0 server, a bioinformatics server testing antigenicity, had a score and produced a score of 0.66, above the threshold of 0.4, indicating that this final vaccine we produced is actually antigenic and can produce an immune response against the rotavirus. Additionally, it's also predicted to be soluble, which is such an important aspect of a vaccine because during administration, it must be able to be soluble within the body in order to produce that response. So here's the protein structure of our Rocio vaccine, predicted by Neurosnap AlphaFold. So using this protein structure, we're able to conduct molecular docking. So molecular docking is a very, very famous technique in the bioinformatics world where it analyzes these simulations between proteins. So it analyzes these interactions of proteins simulated in the body to test it for binding affinity or how well it's able to bind to each other. So you can see in figure A here, we've color-coded our vaccine candidate in as blue for our CD4 or helper T cell epitopes, pink for our CD8 or cytotoxic T cell epitopes, and green for our B cell epitopes. So using each respective portion of our vaccine candidate, we docked it with its respective antigen receptor. So you can see here in B, we have CD8 with its CD8 receptor, C, we have CD4 with its CD4 receptor, and D, we have the B cell epitopes with its B cell receptor. So each of these models were produced by the ClusPro server, and the ClusPro server outputted these lowest energy levels with an average of negative 1,600 around. And that binding affinity score indicates how well this ligand of our vaccine candidate and the receptor is binded to each other, which shows that our vaccine candidate is able to dock and bind really efficiently to these receptors within the body. So additionally, we also conducted an immune simulation of our vaccine candidate. So this provided a bit more of an insight into how the candidate produces cytokines over a long period of time. So the vaccine candidate had both a really robust innate or immediate response, as shown in the second graph, and adaptive, as shown in the first graph, response. So you can see here in our graph, especially, it produced major cytokines, including interferon gamma, right from the very first day, and multiple immunoglobulins after a few days, showing its very strong immune and adaptive response of our Rocio vaccine candidate. So with this project, we're able to simulate and visualize this vaccine candidate through two different vaccine display platforms, including the M13 bacteriophage and the E. coli system. So with the M13 bacteriophage, using the P3 protein, we're able to see that's a very strong tool in terms of displaying our vaccine candidate for epitope presentation. Whereas in the E. coli system, due to molecular docking, as I just explained about, the candidate showed a very strong binding affinity with its target receptors. Additionally, our vaccine candidate, as I mentioned before, it was predicted to have very strong biophysical properties, including antigenicity, non-allergenicity, non-toxicity. It's also strongly hydrophilic, among many other characteristics. So this shows a very, very strong first step into developing this licensed vaccine for the rotavirus. You know, after re-emerging in such recent times, it's really important that we have more research and more research conducted on the rotavirus. So this project provides a really important insight into how we can develop this vaccine purely through bioinformatics servers at home. And we can use this initial vaccine candidate that we proposed into in vitro and in vivo experiments to ensure its safety and stability of the vaccine. So we can also conduct a molecular dynamics simulation which would allow us to get an even more stabilized structure for a rosio vaccine and develop it even further before reaching the lab. Here's my references, and thank you so much for listening.